Researchers from the University of Queensland have made a significant discovery related to the process of aging within human cells. The Queensland Brain Institute's Associate Professor Stephen Zurin and Dr. Michael Dye have unveiled an intriguing role for a protein named ATSF1 that resides deep within human cells. The protein ATSF1 is responsible for maintaining a delicate equilibrium between the generation of new mitochondria and the restoration of impaired ones. Mitochondria, the cellular structures containing their own DNA, produce energy that fuels various biological activities. However, the toxic byproducts of this energy production contribute to the acceleration of cellular aging. In moments of stress, especially when the DNA within mitochondria becomes damaged, the ATSF1 protein takes charge of prioritizing the repair process. This proactive repair mechanism is linked to fostering cellular well-being and extending lifespan. Dr. Zurin likened this relationship to a race car entering a pit stop for necessary maintenance. In their investigations, the researchers observed the effects of ATSF1 on C. elegans, also known as roundworms. Enhancing the protein's function notably enhanced the overall health of the worms as they aged, resulting in increased agility and vitality. While the roundworms did not experience a prolonged lifespan, they exhibited improved health throughout their aging process. This finding holds significant implications for addressing age-related diseases, which often stem from mitochondrial dysfunction. Conditions like dementia and Parkinson's are closely linked to such cellular issues. The researchers believe that this discovery opens promising avenues for promoting healthy aging and assisting individuals who face inherited mitochondrial disorders. By delving into the ways in which cells facilitate repairs, researchers may uncover strategies to prevent mitochondrial damage, which is a critical step toward counteracting the effects of aging. The ultimate objective of this research is to extend the functionality of tissues and organs that tend to decline as individuals age. Dr. Dye emphasized that understanding how deteriorating mitochondria contribute to this decline is key to this endeavor. The researchers envision the possibility of designing interventions that maintain the health of mitochondrial DNA for extended periods, thereby enhancing overall quality of life. These groundbreaking findings were published in the prestigious scientific journal Nature Cell Biology, showcasing the significance of this research within the scientific community.